Hello everyone, this video is to carry out indole production test. So, it is a biochemical test wherein we use tryptophan broth, and this tryptophan that is present in this broth can be broken down by certain microorganisms with the help of the production of tryptophanase enzyme, and microorganisms like E. coli can produce that enzyme, and thereby they are capable of breaking down tryptophan to produce indole. And that indole can be detected when we add Kovacs reagent to it. A ring, cherry red ring is formed on top of it. So, that is what is indicative of indole production. Now, these biochemical tests are basically carried out to confirm a particular bacterial species. Say like E. coli is a bacteria that will be positive for indole. At the same time, another member of the Enterobacteriaceae family, Klebsiella, that will come negative. So, let us see how do we carry out this particular test. So, what we do first we take the tryptophan broth and we place it into tube. Here we will be taking two tubes, one is the test and the other one will be control. So, we need to do it near the flame so that there is no contamination that can take place. We need to cover the cotton plug properly, and we will have to mark both the tubes properly. So, this is our test. We have written T for test, and herein we will write C for control. Now, we will have to inoculate the test organism. So, this is the test organism, the microbial culture, the broth culture that we have. So, we will be inoculating this broth culture into the test tube, but we will not inoculate that into the control tube. So, we will be comparing the result with the control. So, we will have to inoculate the broth into the test tube. We need to heat the loop till it becomes red hot, cool it down, and then take loop full of the culture, take the tube and then inoculate the microorganism into the tube. Mix it well, heat the loop till it becomes red hot so that the microorganism present is dead and it does not contaminate any other culture. And now inoculation is done, and we will have to incubate both test and control tubes into the incubator for 24 hours in 37 degrees centigrade. Tomorrow, after incubation is over, we will be adding Kovacs reagent to both the tubes. If there is indole, then the Kovacs reagent reacts with the indole and it will form a cherry red ring on top of it. If it does not produce indole, then there will be no cherry red ring formation. So, production of indole is indicative of the presence of the tryptophanase enzyme, which has broken down the tryptophan into indole. So, that is what we will be looking for after incubation.